Today we will talk about the monohybrid cross. What is monohybrid cross? This monohybrid cross, it deals with the inheritance of a single character. So when we consider the inheritance of, when we study the inheritance of a single character, we call it monohybrid cross. So it deals with the inheritance of a single character. Monohybrid cross, it deals with the inheritance of a single character. It deals with the inheritance of a single character. As you know that one character, it is controlled by one gene and it has got two alleles. One gene has two alleles which are present on two homologous chromosomes, on a pair of homologous chromosomes. So we can say that this monohybrid cross, it considers the inheritance of a single gene or a pair of alleles, or it considers the inheritance of single gene. It considers the inheritance of a single gene or a pair of alleles. So we can say that how we can define this monohybrid cross. Monohybrid cross is simply a cross which is made between two individuals of the same species. For example, these are two individuals which belong to same species. When we made a cross, when we made deliberate breeding between these two individuals which belong to same species, and then we will study one character. For example, in these plants we will study seed shape. For example, we'll study flower position, we'll study height of the plants. So we can say that this is what is called as monohybrid cross. So monohybrid cross, it is a cross between two individuals of the same species so as to study the inheritance of a single gene, or so as to study the inheritance of a single character. So in case of these monohybrid cross, we have phenotypic ratio. This phenotypic ratio is 3:1. It is 3:1 phenotypic ratio. In case of monohybrid cross, we have phenotypic ratio 3:1 and genotypic ratio is 1:2:1. That is how I explain this. This means that 25% individuals. 25% individuals have 25% individuals have 25% individuals are homozygous. They have homozygous dominant trait, exozygous dominant trait. 25% individuals have homozygous dominant trait. Then we have one, that is 25% individuals homozygous dominant trait. This then 50% individuals have heterozygous dominant trait. They are hybrid. They are hybrid individuals, that is, they are heterozygous dominant trait. Heterozygous dominant trait. And then we have got 25% individuals. The 25% individuals, they have homozygous recessive trait. They have homozygous recessive trait. So in case of how we can explain this explanation, now we'll explain this phenotypic ratio, how we got the phenotypic ratio, how is genotypic ratio obtained in case of monohybrid cross. So Mendel, he crossed the pea plant which is tall pea plant with a dwarf pea plant. So we have parents, this is phenotype, explanation is Mendel crossed pure tall pea plant with the pure dwarf pea plant. Phenotype is pure tall crossed with pure dwarf. Pure tall pea plant with pure dwarf pea plant. Obviously, what would be the genotype? Genotype is simply capital T, and small t, small t. This pure, what do you mean by pure? Pure is homozygous for the trait. Your true breeding individual. It is homozygous or true breeding individual. True breeding individual. That means if, for example, like I say that this is a pure or it is homozygous for the trait, it means that if this dwarf pea plant is self-pollinated and self-fertilized, we get the offsprings which are always have the same trait. They are always dwarf. This is what is called as they are true breeding individuals. Same is the case. This is true breeding. Capital T, it is homozygous. It is pure because it has two identical alleles for the gene. And if we'll take this pure tall pea plant, we'll go for it is self pollination and then self fertilization. We'll find that all its progenies would be tall. That means it is true breeding individual. Then obviously we have gametes. Mendel crossed pure tall pea plant with the pure dwarf pea plant, and then collected the seeds from this cross and raised the pea plants from those seeds. And what he found all the pea plants which were obtained from this cross, they were tall. So he called them F1 generation. So we'll make this cross. This pure tall pea plant, pure dwarf pea plant is their genotype. Then gamete formation. That means law of segregation will operate. Law of segregation will operate here. That means during gamete formation, the two alleles of a gene separate. One allele goes to one gamete, another allele goes to another gamete. So gametes have always one allele of the gene. So we can say that gametes are always pure for the gene. So it would be like this, capital T. One allele will go to one gamete, another allele will go to another gamete. Same is the case, one allele will go to one gamete, another allele will go to another gamete. Then we have fertilization, what is called as random fertilization. Because which gamete will fuse with which gamete, it is a matter of chance. So we call it random fertilization. For example, this gamete will fuse with this gamete, or this gamete can fuse with this gamete. So this is what is called as, we obtained F1 generation. It's called as first filial generation, F1 generation. And here these F1, the plants in the F1 generation, they were all tall. But they were hybrid, or we can say that heterozygous for the trait. Heterozygous for the trait height. So they are tall. They are not pure tall, but they are hybrid. That means hybrid individual. Hybrid individual is obtained when we breed two genetically different parents. We have this is a genetically different parent from this parent. This capital T, this small t, they are genetically different parents. When we go for their breeding, when we go for their cross, we obtain the F1 offspring, that is known as hybrid, because it is having the genes characters of both the parents, although some are expressed, some are not expressed. So this is what we called as F1 generation. So Mendel then did selfing of these F1 plants. So selfing, in case of selfing, the individual is, for example, what do you mean by selfing? In case of selfing, one individual is involved. We, for example, self-pollination and then self-fertilization in one individual, in single individual. But selfing can also take place in a colony having the identical genetic constitution. That means all F1 individuals, they have same genetic constitution, and when we go for a cross between these F1 individuals which are having the same genetic constitution, it becomes selfing. So F1 individuals which have the same genotype, genetic makeup, they were crossed with each other. F1 individuals crossed with each other. So that we get F2 generation. So we have capital T, small t. This individual having genotype capital T, small t, it is crossed with the another F1 individual having the same genotype, that is selfing, capital T, small t. Obviously, gamete formation will take place. This is one allele will go to one gamete, another allele will go to another gamete. Same is the case here. Same is the case here. This is one allele will go to one gamete, another allele will go to another gamete. So then we have fertilization. What is random fertilization? And we get what? We get F2 generation. We get F2 generation. That means this gamete will fuse with this gamete. So this is capital T, this is capital T. This gamete will fuse to this gamete, capital T, small t. Then we have this gamete will fuse to this gamete. We have capital T, small t, and this gamete will fuse to this. We have small t. So this is the genotype. What would be the phenotype of F2 generation of F2 individuals? F2 individuals. So it is tall, dwarf. So here we obtained dwarf. Although the allele, the gene for the dwarfness, the dwarf character was not present in the F1 generation, but we obtained dwarf character, dwarf trait in the F2 generation. It means that the allele, the gene for the dwarfness, it was already present in the F1 generation, but in the F1 generation it has remained unexpressed. But in the F2 generation, this allele, this gene got expressed. So we can say that the allele which expressed in the F1 generation, that is dominant allele, and the allele which had not expressed in the F1 generation, that is recessive allele. So this has this dominant trait, it was unexpressed in F1 generation, but it got expressed in the F2 generation. That is why we obtained dwarf plants in the F2 generation. So obviously, what would be their designation? This is pure tall. Pure tall. These are two identical alleles for the gene. So it's pure. It is heterozygous. This is also heterozygous. What we call as hybrid. This is what is two identical alleles for the gene. We call it pure or homozygous. This is also pure or homozygous because two identical alleles for the gene. This is This is true breeding. This is also true breeding. This is true breeding individual. This is true breeding individual, because this on self-pollination fertilization, this plant will always produce tall pea plants. Same is the case, this is pure or homozygous for the trait, and on self-pollination and self-fertilization, this plant, dwarf pea plant, it will always produce dwarf pea plant. So we call it, it's a true breeding individual. So then what is the ratio of tall is to dwarf? Ratio of how many tall are there? 1, 2 and 3. So 3 is to 1. So it becomes our phenotypic ratio. This is phenotypic ratio. What is the genotypic ratio? Genotypic ratio, we have capital T, then we have genotypic capital T, small t, and small t, small t. How many capital T, capital T? This is 1. How many? These are 2. How many? These are 1. This. So this is our genotypic ratio. Genotypic ratio. For the sake of convenience, this cross can also be represented by means of a Punnett square. It would be convenient to represent it by means of a Punnett square. So we have got this Punnett square. This Punnett square, it was divided by R.C. Punnett so as to show the results of cross between the two individuals. We have F1 individuals, they were crossed with each other. These are F1 individual, these are crossed with another F1 individual. So these are F1 individuals were crossed with each other. F1 individuals were crossed with each other. So this cross, obviously gamete, this male gamete, this female gamete formation will take place in this individual. There are 2 alleles of the gene, so one allele will go to one gamete, another allele will go to another gamete. Capital T, small t, capital T, small t. So we have capital T, capital T, capital T, small t. This individual be having genotype capital T, small t. This individual be having genotype small t, small t. So this is pure tall. This is hybrid tall. This is hybrid tall. It's pure tall. So same, we have 1, 2 and 3 tall. 3 is to 1 phenotypic ratio. And what is genotypic ratio? We have 1 pure, 2 hybrid and 1 pure dwarf. So this is phenotypic ratio, this is genotypic ratio. So these are identical. For the sake of convenience, we use the Punnett square checkerboard to show the results of a cross between the two individuals. So this is actually Mendel, he observed the same results in the other traits. Here he took the plant height, that is one character. Then pure tall pea plants were crossed with dwarf pea plants, and in the F1 generation tall plants were obtained. Then he inferred that the character which gets expressed in the F1 generation, the trait which gets expressed in the F1 generation, that is a dominant character, that is a dominant trait. And the trait, the character which remains unexpressed, which does not express in the F1 generation, that is a recessive trait, that is a recessive character. He observed the same results with other characters, with other traits. For example, seed shape. We have pea plants with round seeds, pea plants with wrinkled seeds, and in the F1 generation he obtained pea plants with round seeds, and he said that round seeded pea plants, round seeded, it is a dominant trait over the wrinkled seed. Same is the case, pea plants having yellow seeds that were crossed with the pea plants having green seeds. And in the F1 generation, he observed pea plants with yellow seeds. He said that yellow color is a dominant color over the green color. Same is the case with the flower position, pod shape, pod color. And from this, he got the idea of that a character has two contrasting forms. One is dominant, another is recessive. And he was lucky to find these contrasting forms, dominant form and recessive form, in his hybridization experiments. Then this monohybrid cross, it was also useful for him. He also deduced the law of segregation from the monohybrid cross. So this monohybrid cross, it was useful for him to deduce the law of segregation. Law of segregation. That means at the time of gamete formation, the two alleles of a gene separate. One allele goes to one gamete, another allele goes to another gamete, and their fusion is a matter of chance. So this is a law of segregation which was deduced from the monohybrid cross. So we discussed what is monohybrid cross, their ratio, phenotypic ratio 3:1, genotypic ratio 1:2:1. Thanks.